Okay, I don't know how this is going to work or how well this is going to turn out, but I can guarantee you it's going to be much easier than me emailing you and explaining to you the whole concept of the arena life cycle. Okay, this is how I taught Sam. This is also how I kind of imagine or think of it myself. So I'm just going to start off with going through DNA, for example, because DNA is Ambisense. And this is, I think, one of the things that people are having trouble picturing. So anyways, so if you look at this, so here's our DNA. We got our two strands, the complementary strands, any parallel, yada yada yada. The reason why it's Ambisense is that each strand within itself can code for a protein. So each strand can be used as a template to transcribe mRNA, which then in turn can be translated into protein. So focusing on this one first, on the top strand. Actually, one thing to know and keep in mind is this is important, all polymerases use a 3' to 5' template to create the proper 5', 3' transcript. Okay? So remember that. So just starting with that, establish that polymerases use 3' to 5' template to make 5', 3' mRNA transcript. So in that sense, with respect to that, you see here that the bottom strand is just the coding strand, and you see it also has encoded in that strand is a start codon sequence, but that's for later. Focusing on the top strand, which is the 3' to 5' template strand. The template strand, so that the polymerase will read at 3' to 5' and transcribe that into an mRNA transcript. And you can see there, I'm showing here that GAC, that's the sequence within this strand that will be, when transcribed, become transcribed into a start codon for the particular ribosome. Okay, so that's just focusing on the top strand, because I'm just trying to separate how two strands can make, or these two strands each can code for a particular gene. Each strand can code for a protein. Now, if you flip this around, kind of just like this, or not even just flip it around, just in the sense that now we're focusing on the bottom strand. Flipped around in the sense now the bottom strand becomes a template strand, and the top strand is just a coding strand. So the bottom strand is the template strand because, again, within this strand you have a DNA sequence which, when transcribed, will become a start codon sequence on the mRNA transcript. And template strand, in that, it's going to be read from 3' to 5'. So the polymerase reads this from 3' to 5', starting from the start codon, and it'll transcribe it into the mRNA transcript. It looks weird here, but another thing to keep in mind, this is the second point, is that ribosomes synthesize proteins 5' to 3'. And that's why I had to write it this way, AUG this way, because it has to synthesize, the ribosome is actually reading it this way. And if you see here, the ribosome is reading it this way. So I really hope that makes sense for now. Just a couple things to add. Just also, you can think about it that each strand for DNA has 3 reading frames, right? So technically, each strand, because it codes for 3 reading frames, that includes a total of 6 reading frames. And I just realized that's irrelevant. Just forget the whole. I tried to make it more complicated than it is. Anyways, so that's the whole concept of Ambisense. You cannot think of Ambisense as with respect to one strand. Ambisense is referring to 2 strands. Ambisense in the sense that each strand can code for a protein. Okay, so I hope I got that first cleared up. Hold on, I brought the wrong thing. One sec. I'm going to actually end this video and then send you another one for the actual arena virus because I brought the wrong diagram.